Hello guys, welcome back to Learning with Isha. In the previous videos, we have already covered monohybrid cross and Punnett square. Today's video is going to be one step ahead. It is dihybrid cross. We are going to use the Punnett square technique to find out our answers without any confusion. That was the purpose of the Punnett square. So let's begin. What is dihybrid cross? Dihybrid cross is a cross where we study the inheritance of two characters simultaneously. मोनोहाइब्रिड में हम क्या कर रहे थे, टॉल और शॉर्ट, लेट्स से ये कैरेक्टर्स ले रहे थे. डाईहाइब्रिड में हम क्या करेंगे, एक ऐसा पेरेंट लेंगे जो कि टॉल भी है और उसके सीड्स राउंड भी है. तो मेंडल ने 7 कैरेक्टर्स स्टडी किए थे, उसमें से हम कोई 2 पिक कर लेंगे. ओके, सो हियर आई हैव चोजन टॉल प्लांट विथ राउंड सीड्स and another parent is a short plant with wrinkled seeds. Now this depicts the phenotype of the parent generation. I have already listed it. Now let's assume this is the male parent and this is the female parent. So we know the phenotype. Now let's try to write the genotype. अगर tall plant है, ये एक dominant trait है, capital letter से denote करेंगे. Short एक recessive trait है, small letters. Okay, now let's move on to the next character. Round seed dominant है, capital R से. Wrinkled seed recessive है, small r से. This is the genotype of the parents. Next step क्या है, to list down the gametes. So let's make the gametes now. The gametes will be T and R. T and R, because there is no other, it's a homozygous parent. So all the gametes formed will be of a single type. Same is the case over here. So all the gametes are of the same type. Now what is the next step? Next step is to cause a fertilization and form our F1 generation. Normally what we do is we draw lines. Now that we know how to make a Punnett square, let's write it with a Punnett square today. So we are making the F1 generation via a Punnett square. So how will I do it? First, I'm going to draw a square. Okay. Now, usually I list the male gametes on the left hand side. I'm just quickly going to do that. T, R, T, R. This represents my male parent. The female parent is listed on the upward side. Small t r, small t r. Now what do? We fill in the blocks and the blocks represent the genotype of the offsprings. Let's quickly do that. Okay, I hope this is the genotype that you obtain for all the 4 offsprings. Now here we see that all the 4 offsprings have the same genotype. So the genotype is capital T, small t, capital R, small r. This is for the F1 generation. Now this denotes a tall plant having round seeds. Tall dominant hai, round bhi dominant hai. So we get a tall plant having round seeds in the F1 generation. Now if I ask you the genotypic and phenotypic ratio for the F1 generation, it is going to be what? All plants are of the same type. तो एक ही जीनोटाइप पॉसिबल है ये, तो क्या हो जाएगा, 4 on 4 आपका यही हो जाएगा. Similarly for the phenotype, तो यहाँ पे जीनोटाइपिक और phenotypic ratio don't matter much because all the 4 offsprings are of the same type. Now in the next step what we do is, we take this F1 offspring and perform what? A selfing, a self-pollination step. So now let's quickly move on and do the selfing. Now we know that the F1 generation plants are going to be my new parents. I'm going to get an F2 generation. Now I know कि मेरे जो parents है F1 generation में, उनका ये genotype है, ये उनका phenotype है. The next step is to make the gametes. How am I going to do that? Now ये important part है in a dihybrid cross. आप first step में क्या करिए, आपका जो height का trait है, उसके दोनों एलील्स को सेपरेट कर लीजिए. सिमिलरली फॉर द अदर कैरेक्टर भी आप एलील्स को सेपरेट कर लीजिए. ओके, नाउ व्हाट यू आर गोइंग टू डू इज सिंपली ड्रॉ दोज लाइंस जो मोनोहाइब्रिड में करते थे, और क्या करेंगे, डिफरेंट कॉम्बिनेशंस को लिस्ट डाउन कर लेंगे. लाइक लेट्स क्विकली डू दैट. कैपिटल टी के साथ पहले कैपिटल आर आ गया, कैपिटल टी के साथ स्मॉल आर आ गया, स्मॉल टी के साथ कैपिटल आर आ गया, एंड स्मॉल t के साथ स्मॉल r आ गया। तो हम बेसिकली चारों कॉम्बिनेशंस को लिस्ट आउट कर देते हैं। नाउ द नेक्स्ट स्टेप इज टू परफॉर्म अ फर्टिलाइजेशन। सो मेल और फीमेल तो सेम ही प्लांट था, सो वी आर गोइंग टू लिस्ट डाउन द गैमीट्स इन द Punnett square that I have already drawn. So let's do that. Capital T, capital R, capital T, small r, small t, capital R, small t, small r. This is for the male parent. Now I'm going to quickly repeat this for the female parent also. And now capital T, same process on the other side. Okay. So this is for my female parent now. So now what I will do is, I will list down the genotypes of the offspring. I have already told you that in a Punnett square, each box represents what? The genotype of an offspring. So naturally, अगर मैं पूछूं F2 generation में how many offsprings will be formed? 16 offsprings, right? The number of boxes, उतने ही offsprings बनेंगे. So how do we fill this now? See, you draw a line from here and from the top, both ways, and just list down whatever you get. Capital T, capital T, capital R, capital R. Now let's say I want to fill this box. What I'll do is just draw a line from here and here. Preferably it should be an imaginary line in your mind. Initially you can draw it like this also. Small t है यहाँ पे भी small t है, capital R and capital R. So now what I'm going to do is quickly fill out the boxes and meet you in the next part. What I've done for you is I've quickly filled in the boxes, written the genotype. Additionally, I have also mentioned the phenotype. This is for you to do. Let me discuss how I've done that. यहाँ पे capital T, capital T, capital R है. क्या होगा? दोनों dominant है. आपका dominant character will be expressed. The plant will be tall and round. Now in this case, we have capital T, small r, small r. So wrinkle trait आएगा in a tall plant. Now let's see here what happens. Small t, capital R. So short होगा, but ये dominant trait है तो round होगा. In the last case, what is happening is small t, small r. सारे ही alleles आपके क्या है? Recessive type के. So the plant will be a purely recessive trait expressing plant. So now what my next step is going to be, after I have filled out the genotypes and phenotypes, I'll try to find out the phenotypic ratio. For that we need to see what are the possible phenotypes present. इसके लिए combinations देखने पड़ेंगे. जैसे कि there could be a tall plant that could have a round seed. ठीक है, आपका एक tall plant हो सकता है जिसमें हो सकते हैं wrinkle seed. Similarly, a short plant having what? A round seed, and a short plant having a wrinkle seed. So these are the 4 combinations possible. इसको कैसे verify करेंगे? We'll verify it through the results of this Punnett square. Now what I've done is, in the blue color, I have written down all the phenotypes that are of a tall and round plant. उनके genotypes ये वाले हैं. Let's see, tall, round. This is also tall, round. See, all the blue ones are tall and round plants. Let's count how many of the plants are tall and round. So 1, 2, 3, 4, 5, 6, 7, 8 and 9. So 9 plants have tall and round characters. Now let's move on and count the tall and wrinkled ones. So ये है tall, wrinkled. 1, 2 and 3. So 3 plants have tall plants with wrinkled seeds. Now the next one, short and round. So denoted by the purple color here, 1, 2, 3. So 3 plants of this type, and there's going to be only 1 plant present in the last block that has tall, short but wrinkled seeds. Only one of this type. So 1. Now, how do I verify if my results are correct or not? I'm just going to add this up. And what should I get? I should get 16. Why? Because I had 16 plants. So, phenotype kuch bhi ho, the number of plants has to remain same. So, 9, 3 plus 1, yes, 16. So, my results are correct. This is a good way of verifying that have I missed out any phenotype or any plant. So, now we already know the phenotypic ratio. I've clearly listed them down. 9 is to 3 is to 3 is to 1. Now let's find the genotypic ratio. How I'm going to do that? Simple है. Tall and round plant. इस टेबल में मुझे पता है कि 9 केसेस ऐसे हैं जहाँ tall and round plants exist करते हैं. Now I have to find what are the different genotypes that represent a tall and round plant. So a simple way of doing it is कि आप first row में देख लीजिए. ये आपके tall और round plants का genotype है. So TT, RR, capital T, capital T, capital R, small r, capital T, small t, capital R, and capital T, small t, capital R, small r. So these are the 4 genotypes possible जो कि आपको tall और round plant देगा. Now you see there's another way of doing it as well. Tall plant है, so how can I denote a tall plant? Either it could be homozygous tall or it could be heterozygous tall. Fine. I'll just list that down. Now the other trait is round. It could be homozygous round or it could be heterozygous round. I'm just going to list down the combinations. Homozygous वाले पहले, heterozygous वाले बाद में. Tall से करते हैं. TT, capital R, TT, heterozygous round. Then the heterozygous tall के 2 cases, उसमें homozygous round and heterozygous round. So this is another way you could go about doing it. Now what I'm going to do is count किस type में कितने plants fall in करते हैं. So see, this is TTRR, so 1 over here, 1 over here, 1 over here, okay, here 1, this one is of this type. So what I'm going to do is simply count in this way and keep marking lines. Similarly, I'm going to repeat this process for the other traits as well. Tall and wrinkled. Tall and wrinkled के मुझे पता है 3 plants है. इसमें ये हो सकता है homozygous tall और heterozygous tall. Wrinkled क्या है? एक recessive trait है, तो एक ही type के इसमें आपको alleles मिलेंगे. So there are only 2 combinations. You see here homozygous tall and homozygous. Recessive though is always homozygous only. So 1 for here. Similarly here 1 line I'll draw here and this one falls in this category. So just count and write 1 plant of this type, 2 plants of this type. Repeat the process for the entire genotypes listed down here and what we'll get is the genotypic ratio. So after listing down, after all your counting is complete, you should come out to such a number. 1, 2, 2, 4, 1, 2, 1 and 2, 1. This is what? This is your genotypic ratio. So it is 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1, 1 is to 2 is to 1. This is my genotypic ratio. My phenotypic ratio was 9 is to 3 is to 3 is to 1. Now how do I verify that my genotypic ratio is correct? I'm just going to quickly add up these numbers and it should add up to a total of 16. So 3 to 5, 9, 10, 12, 13, 15 and 16. So this verifies that my genotypic ratio is correct. Do let me know in the comment section below if you are interested in watching another example of the dihybrid cross. The next video is going to talk about back cross and test cross. In the further videos, I would also cover a tri-hybrid cross. Thanks for watching to the video. Please subscribe to the channel to be updated about more such videos. Thank you.